This is a production of Cornell University.
Thanks, it's a great pleasure to be here. Can you hear me okay? Yeah, good. Okay, so yeah, I'm really fascinated by size. Size is really important and it's a deeply fascinating question. Size is fundamental to everything and it's really important for yield. But size is not trivially important to yield. You might think we want things to be bigger, but we only want the right part of the plant to be bigger. We want the grain, for instance, or the part of the plant we're going to eat to be bigger, but we want the rest of the plant often to be smaller to fit into more plants into the same field or the same growing space. And so if you think back to the Green Revolution, one of the key things was making smaller plants with bigger grains. And here you can see pre-Green Revolution wheat is larger plants, smaller grains. Post-Green Revolution, you've got smaller plants, bigger grains. So we need to understand size. Breeders are very good at achieving this, but I think that if we actually understand how size and shape are controlled, we can do even better. And size is one of the things we really still don't understand that's so basic. So my lab studies this in Arabidopsis. We like the Arabidopsis sepals and the leaves for studying this. So our sepals are our green outermost floral organs. They enclose and protect the flower bud, so they're really defensive organs. And one of the reasons sepals are cool is that they have this amazing pattern of different cell sizes in them in Arabidopsis. So they've got these giant cells, false colored here in red, interspersed between smaller cells. Another reason we chose sepals is just that they're on the outside of the flower, so they're really accessible for imaging and manipulation. Another reason is that there's 4 sepals in each Arabidopsis flower, so we can look at their size within a flower and say, are all the sepals the same size or not? And that's actually important for their function. The sepals need to be the same size to keep that flower bud closed and keep all the environment, insects, everything out while the reproductive organs are developing. So sepals are a great model system. We've now gone on to leaves, which are totally not a great model system, but a lot of people care about leaves. And guess what? Sepals are like little leaves, so that makes me happy. Okay, so what we love to do in the lab is actually image the plants as they're growing and try to understand how the growth and division of the cells is giving rise to the size and shape of the organ. And so here we've got living plants, and this is what I did back in the day, taped onto a slide, and I've imaged it every 6 hours and watched those sepals grow. So this is a flower bud. You can see here's one sepal, here's one sepal, here's one sepal. And what you're seeing is the fluorescent plasma membrane marker outlining the cells and fluorescent nuclear marker. You can see there's all kinds of complexity going on here. These cells are not doing simple things. Some cells are getting really big nuclei and growing a lot. Other cells are dividing and making a ton of smaller cells. And all of this is coming together to coordinate so that the sepals are growing at basically the same rate, despite the cells doing all kinds of things. Now I'm going to continue the theme of image analysis and say that we love to understand this computationally through computational image analysis. This is done in collaboration with Richard Smith's lab with their amazing morphographic software. And I'll play that again, hopefully. So we've colored each cell and all of its daughter cells with the same color. It doesn't want to play. And so you can see that, okay, there it jumped. One green cell gave rise to this whole patch of green cells, and one red cell gave rise to this whole giant red cell. And so that's how giant cells are forming. They're growing and not dividing the whole time, whereas other cells are dividing. So we can measure the growth rates, we can measure the growth directions, we can measure all kinds of things from these images, and also gene expression patterns. And so we're trying to put all of this together to understand how this organ is growing. Now we do a lot of molecular genetic work that I can't possibly show you in this 8 minutes, and come up with gene regulatory networks. I don't expect you to understand anything about this, except that there's genes interacting with each other, giving rise to something. And that something we like to model. And so we collaborate, this is with Pau Formosa Jordan and his postdoc Nicholas Russell, to model the growth and division of these cells with their decision-making process based on this gene regulatory network about whether they divide or grow. And you can see we've got proteins fluctuating up and down, and what's coming out of this model is a pattern of different cell sizes, a bit like a sepal, where we've got giant cells, seen here in purple, interspersed between smaller cells in brown. So you can see it's a model, give it credit, it's not quite like a sepal, but we're still getting that pattern out of our gene regulatory network. One of the reasons I really love modeling is not when it works like this does, but when it doesn't work. When it doesn't work, it tells us what we're missing. We're like, oh, wait, we hadn't thought about this part, or we don't have it quite right yet. Those never get published, but they're the most important models, actually. So my lab is really focused around two major questions. One is, how are cells of different sizes created during development? And we're really looking at this, how do you form giant cells in the sepal and the leaf? Both have giant cells. And we found this really interesting role for stochasticity or randomness in this process. We think about development as being deterministic, everything's programmed in, but actually the plant in this case is using randomness to generate this pattern. So that's exciting to us. The second question is sort of the flip side of this. You've got all the cellular variability. How are organs made with reproducible size and shape, despite variability in the cell growth, the cell size, cell division, gene expression, all of these things? And so if you look at across these Arabidopsis flowers, you can see they all have 4 sepals. All the 4 sepals in each flower are the same size. And they're the same across flowers. So how does the plant do this? How does it make this reproducible structure from this mess? And then we want to apply this to engineering living materials. So we have recently got a grant from the National Science Foundation and a big collaborative Biology Integration Institute, 12.5 million over 6 years, with a huge collaboration of people led by Pam Soltis at University of Florida to study polyploidy, because our giant cells are polyploid. They have more than one copy of the DNA. So we want to look across organisms and understand, what does having more copies of the DNA do to the cells? How does it make them bigger? What other properties does it have? And then we also are very interested in applying this to engineering living materials. Our materials are not sustainable, and we'd like to actually use the properties of living organisms, specifically bacteria, fungi, and plants. They have amazing properties, and can we incorporate them into our materials, into our buildings, into even our chairs, into our clothing, to make them better? And think about what all of these organisms are good at. They use non-petroleum-based energy sources. They can sense, they can adapt, they can repair. Think about our roads. What do we hate about our roads? All these potholes. Roads cannot self-repair. But what if we incorporated living materials into them so that they can self-repair? How awesome would that be? Okay, that's a dream at this point, but it's a dream we've been talking about. Lori Huberman is part of this Engineered Living Materials Institute as well. And so there's a bunch of challenges. And speaking to the plant path group, I think one of the big challenges is, how do we incorporate different organisms interacting with each other? Because we know we're going to have many ecosystems to do this. The challenge that most fascinates me is, how do we engineer the shape and structure and mechanical properties and growth of these organisms? And we're starting this with a bit of diatoms, because they have silica-based frustules and they're self-organized nanomaterials. So look how cool these little structures are, how they can make them at room temperature. This is work by my postdoc, Trevor Cross. And that's it. I'd like to thank my lab for all of this inspirational work. We could not do this without amazing collaborators, both at Cornell and beyond. And thanks to NIH and NSF for lots of funding. So happy to take any.
Thanks, Adrienne. Any questions?
You showed some models, and I wonder how many parameters do the models that you play with have?
How many parameters do the models we play with have? We try to keep the parameter number as low as possible, because you can get anything to happen in a model if you add enough parameters. So we're really trying to limit it down to things where we have some kind of biological basis. That said, there is some level of parameters you have to have, and most of the time we have no biological information for those parameters. So we do a lot of parameter optimization and look at in what regime do we get the behavior we expect, what regime do we get other things. And so there's a lot to be done there. But yeah, we're trying to keep the parameter number low.
For giant cell, giant is a consequence of polyploidy, or are giant cells polyploid consequence of the giant?
That is a question I've been wondering about for years. And they go hand in hand, and we so far cannot separate those two things. And I think that's one of the things I would love to do is separate. We need something that actually separates ploidy from size, and we don't have it, which tells you what a universal principle that is. So the question is, what's cause and effect? And it's totally unclear at this point.
Follow up to the pathology crowd here. Is there anything we learned about the development of feature from nematode pests that create giant cells, syncytia in the root system?
Yeah, so those giant cells are unrelated to these giant cells. We actually sent the molecular marker for these giant cells to a group that was working on that, and yeah, it didn't show up. So that was disappointing slightly. One of the things I should say about these giant cells is that they are little biosynthetic factories for a lot of defensive compounds, and our plants that overexpress giant cells are actually more resistant to things like powdery mildew, and our plants without giant cells are little farms of powdery mildew. So there is some defense response going on here that's really interesting. So thank you.
Good. Hi, everyone. My name is Hale Tufan. I'm an associate professor in plant breeding and genetics. And just like Justine, even though she didn't mention it, I started with lightning with an E. So I thought, okay, I'll just use lights in my title slide. So I'm going to give you a lightning talk about our work. And it's a bit strange to be here because I actually did my PhD in molecular plant pathogen interactions. And what I do now is very different. So in a parallel universe, this would be my department. So it's nice to be home. So I'm going to start with a premise. This is very busy, but I'm going to give you the bottom line. And that is our agri-food systems are driven by structural gender inequality. What that means is from value chains producing, to distributing, to the food environment, to consumer behavior are all shaped by societal structural inequalities. Okay, this is pretty well established at this point. What we also know is how women specifically engage in these agri-food systems globally is shaped by their agency, social norms, their access and control over resources, and so on and so forth, right? So this schematically tells you there's a problem, right? There's a global gender inequality in agri-food systems, which then shapes outcomes from how people engage in those agri-food systems. Okay, bear with me. I'm giving more intro slides because it's going to be a little bit different from what you watched. So the most recent thinking in this space says, then if there is a problem, how do we rectify it? How do we change it? What are the levers of change towards more equitable agri-food systems? Tells us that we can look at individuals engaging in agri-food systems, households, communities, and so on and so forth, right? There's different layers where you can affect change to really rectify the inequalities. And these look at institutional inequalities, power relationships, agencies, and social institutions, okay? So we're gonna come back to this. It's like a crash course in gender inequality in food systems. Within that system of agri-food systems, innovations, agricultural innovations are a subcomponent, right? We innovate, we create new varieties, we do mechanization so that we make farmers' lives easier, right? But what we know now is, again, the latest thinking is innovations don't exist in a vacuum. You don't just develop a tractor and airdrop it, right? There has to be more give and take with the communities who are going to adopt it. So the latest thinking is around context-dependent socio-technical innovation bundles. Say that 3 times very quickly. What that basically means is as you're innovating for an agricultural technology, you must know where that technology will take place, right? Where will it, how in people's lives will it enter? How will they use it? How will they interact with it? So you really need that context dependency. And that's where we are. So by we, I mean the EQUAL Lab. We call ourselves the Equitable Agricultural Research Lab. We just set up last year. And really we take an agricultural innovation, which is crop improvement. That's end to end from a design of a plant variety all the way to the release and seed systems. And we say, how can we really look at gender equality and social inclusion in that whole process, okay? Now, because these are complex problems, we can't just do it unilaterally. We need a group of people from very different backgrounds. So our lab is kind of a hodgepodge of different disciplines, and we all kind of spend half our time trying to communicate with each other. But that's the only way we can solve these complex problems, right? So we have people who have plant breeding backgrounds, economists, anthropologists, and we all come together, and I'll give you examples of what that looks like. We're not tethered to a geography. We engage in a lot of different projects. So the sites of the research tend to be very different, as well as crops we work on, because it's agnostic to a degree. These inequalities are pervasive, right? They're not in one context. But what brings us together is how we research, right? Using feminist research networks, frameworks and approaches, and a lot of trans or interdisciplinary methods. There's not one solution to these problems. And we do a lot of collaborative research. None of this happens on our own. We're constantly communicating, working with programs globally. So to sit this on some sort of organizational principle, like you just seen, it's not a mess, it's just range. So the way I look at it is we do work on data generation, which basically means we want to understand what people in different parts of the world and different social heterogeneity look for in crop varieties, right? That's what I call data collection. Traits, varietal preferences, how do we collect that? We also do methodological work that says, how do you collect data in a way that's more inclusive? I'll talk about that a little bit. We try to synthesize that up, so we're giving the right messages to the plant breeding programs to say you should care about this or that. And then we do more analysis of the research itself. That's the more fun part, in my personal opinion. I don't have fancy videos, so I'm feeling a little disadvantaged, but I'll show you some examples of our research. This may be very new, and you're probably thinking, okay, what does this look like? What kind of research does this culminate to? So I'll give examples of each. On the left, both of these graphs are on cassava in Nigeria. So we do a lot of work with breeding programs there. And the question the breeders often ask is, who wants what? What traits matter to whom and why? What varieties matter to whom and why? It seems easy, very difficult, right? So in years and years of data have accumulated on what farmers want from traits. But what we also know is that farmers are not homogeneous groups, right? They're very different. Where they are, what kind of households they're living in matters. So on this rather busy plot on the left, we're trying to pull apart trait preferences for these traits on the bottom through a heat map here that we're showing it. If you're a woman or a man living in a poor, non-poor household, food secure, insecure, 4 different regions, you may get different patterns of trait preferences, right? Breeders don't like to see this, but we like to tell them it matters. Context matters. Social heterogeneity matters. And this is for trait preferences. What you see here is what's called a placket loose tree. And all this is telling you is these are varieties in rank order. Depending on if the farmer is a man or a woman, and if they're interacting with a man or a woman to process, you get different varietal preferences. What does that mean? We don't know. But what we know is it happens. Because again, people don't live in bubbles, right? They interact with each other, and they may have different preferences because of those social interactions. So the bottom line is we're trying to move away from like these homogeneous trait buckets and really try to understand social heterogeneity, right? At the individual level. Moving a little bit up, one thing that I've become really interested in is methods. So what this means. So imagine you're going to a household, you want to ask people, what kind of cassava do you like? How do you do that, right? That itself is a big question. Often it's surveys, but then you have to ask yourself, who's responding to the survey? There's a lot of power in politics and who takes that survey. So on the left here, you see a kernel density plot where we're asking husbands and wives from the same household, the same question. How many cassava varieties do you grow that are improved? And then we go to the field and we take the leaf samples and we compare them, right? What you see is nobody's consistent, right? The husband's responses and the wife's responses differ from each other, and they both differ from the actual leaf tissue. Now here, I don't want to say anyone's right or wrong, because when we go and do this and we ask people at the plot level, you get a different picture. My point here is who you ask and how matters hugely for adoption rates. Our assumptions around a variety is adopted or not may be based on wrong assumptions. Who you ask matters hugely, right? This is work in Senegal and Cowpea where we had 5 different traits, and we asked, again, husbands and wives for their relative preferences. Up here, classic, we separate men and women, look at the relative importance, the higher the more important. But then what we did was we took the same couples and we classified them by agreement and disagreement on who makes decisions in the household, right? So then we're saying, are there some households that are more kind of together? And are there more households where there's more disagreement? Does that matter for the choices they're making? Readers don't like us when they see this because it does matter, right? But again, we're trying to tease apart to say, how can we ask these questions so you get good information? Ooh, I ran out of time. Okay, this is a scoping review we did last year, just to tell you, we looked across all trait preference papers globally for all crops. And we asked the question, how are people collecting information? And what you see is it's often direct questions. So when someone gets asked, what do you like or not? And often we don't have data from women. That's the gender bias in the totality of the data is significant. So we don't actually have data on women's trait preferences. Joss is staring at me, so I'm gonna wrap up here. This is the highest level work that I think is really exciting, where we turn the lens back on the researchers themselves a little bit. And we say we are social beings engaging in research. We're not robots, right? We have characters, we have histories, we have social identities. How does that shape how we're actually doing research? There's a lot of theory out there that shows that's true in other fields. So we have two sets of work coming here, looking at care responsibilities of faculty. How do faculty balance care responsibilities and their social identities? How does that play out? And the second one is management and leadership. What does it mean to be a woman in an international agricultural research ecosystem and for their management abilities and perceptions? So this is a much higher level thing. So I want to thank my lab. There's too many names to name here, and I try to put them on the spectrum of work that we do. And thank you for your attention.
Any questions, Pathology?
About like trait preferences, are you able to control for their starting point in terms of like what varieties are growing, or even like soil status?
Interesting. That would be good to look at. We don't often look at soil nutrient status, and this is, I think, where disciplinary practice comes in. Often these questions are asked by economists. If you look at globally how the data is collected, economists do these surveys a lot, and often it's just, I ask you the question, I have a box, and I tick it, and I walk away. What we do control for is you're running kind of like a regression. You look to see if there's any covariates that are actually explaining some of that variability. So we do check if there's things that are bigger than the actual trait preference itself, if that makes sense. So yes, we do check that as much as we can.
I have a quick question. So you're generating data for trait preference as a snapshot, right? So
Yeah.
Contemporary. Do you have any way of generating data on a trajectory historically and forward?
For the scoping review, we tried to do that for 50 years, all crops, to see what are the trends in the trait preferences. The biggest problem is ontologies. Every study will define a trait in a different way. And so extracting that and relating them to one another is hugely difficult because there's no standard for social research on traits. You say yield, I say root size. Yeah. Someone has to decide those are equivalent to look at the totality of the data set. It's something we're working on right now to be able to synthesize it.
Hey, everybody. It's great to be here. I am happy to tell you a little bit about my research and what I do today. I wanted to start off with a first slide. Let's see, let's get there. Here we are. This is a photo taken outside just yesterday. In this photo, you can see the Tower Road lined with trees. And I spend a lot of time thinking about how we interact with plants in this kind of day-to-day level outside of these agricultural systems. And in particular, I focus on the health-relevant interactions that we have with these plants. This can include everything from the services that these trees provide in terms of cooling on a hot day through both shading and transpiration, through to the removal of air pollution, such as particulate matter from the air. There's also a lot of work that shows really extensive mental health benefits through exposure to green spaces and all sorts of other ecosystem services that are also health relevant. At the same time, we have all of these negative interactions with plants. everything from the release of allergenic pollen to the release of volatile organic compounds like isoprene, which is a precursor to ozone formation. Even in the most urban areas, we still have landscapes that are filled with plants. For example, New York City is currently 22% of it is forested. About a year ago now, there's was some local legislation passed to try and increase New York City's canopy coverage of trees up to 30%. And one of the major justifications for this was people said, hey, trees really improve public health, so let's have more of them. This is not just isolated to New York City, and we're living at a time when cities across the United States and the rest of the world too are also channeling resources into planting trees, and a little bit less into maintaining the trees that they already have, much to the chagrin of urban foresters and other urban tree professionals. But it is, right now, there's about $2 billion being put into urban forestry in the US through the Inflation Reduction Act. However, even though one of the major motivations for this is improving public health through making there be more trees, we don't really have a good understanding of how many of these trees, how the individual trees interact with individual people. And so we have a lot of these large scale patterns like, hey, if a neighborhood has a lot more green space in it, it tends to be cooler. But when you get down to the level of how is this individual tree transpiring? How is it providing shade? We don't have very good information at that granular scale, which is a problem because those are the scales at which we make decisions. We don't tend to say like, oh, we're going to change this neighborhood. We say, no, we're going to plant a tree right here. And then we make decisions about where that tree is, what type of tree it is, and so on. And so my goal and the work that's being done in my lab focuses on quantifying these interactions with plants in order to improve the decisions that we make, often around tree management and so on, but also around our behaviors as people too, in order to improve the health of people as individuals and as a broader group as well. So I'm just going to walk you through what this looks like, although you'll have to excuse me because this is a little bit of a listicle right now. Sorry, everybody. So focus on me, not the boring text. So one of the major projects that I have right now is looking at allergenic pollen concentrations, and I'll tell you a little bit more about that in a moment. Another major project that I'm working on is trying to understand the cooling effects of trees in New York City on an individual tree scale in order to understand what heat exposure is like, what the health consequences are. In other words, how does that affect the number of people who are going to the emergency department during heat waves? And ultimately, to be able to inform decisions as we're making all of these, doing all of these urban greening campaigns. Like when you plant a tree, it lasts for a long time. And so we really should be figuring this out now because the consequences of today's decisions are going to last with us for decades. As part of this project, I'm doing a lot of work with a postdoc in my lab, Dave Miller, trying to use remote sensing to identify individual trees. And because you can't do a good job of managing what you don't know. And so we're using some new approaches, incorporating the timing of various life history events for trees, things like when is it leafing out? What color does it look like in the fall to try and untangle this a little bit better? So really using the ecology to inform some of the remote sensing analyses. Some of the other projects that are happening in the lab now and touch a little bit on some of the associations between things like neighborhood income or race and ethnicity, and how that is associated with what trees are where. And there end up being, it seems, large health consequences for some of these things, as different types of trees provide different services. So those are a few of the projects that are happening in the lab right now. And for the last little bit, I just want to walk you through an example of what one of these projects looks like. And so come with me to Texas now and take a look at ash juniper, which is one of the most common trees in Central Texas. As you can, the gray things you see here are not clouds in the sky. That is pollen being released from this tree. Just to give you some context here, people begin to experience allergic reactions to this pollen at concentrations as low as 10 or 20 pollen grains per cubic meter of air. A single pollen cone on one of these trees has about 500,000 pollen grains in it, and you might see up to 1 million pollen grains, or 1 million pollen cones, excuse me, on one of these trees. And so what we're trying to do is to understand both what the health consequences are through epidemiological analyses, as well as to create operational pollen forecasts. It turns out that most of the pollen forecasts that you might find through your weather app, they're total garbage. They're just like unvalidated, like completely, well, I'll pause for right now on it, but there's a lot to be said. I'll hold myself back. And so some of the approaches that we take are doing field work, so field campaigns to take manual measurements. We also work with citizen science organizations, especially the National Pathology Network, to enlist people to help us take these measurements. Then we also do a lot of scaling up to try and understand what is happening with individual plants, and then to take that up to a site level and ultimately to regions or across an entire state, and someday I hope across the entire country. Some of the processes that we try and understand are fairly simple, like how much pollen is a plant producing? And so, Justine, I'm really excited to think with you about some of the methods that you've been using, because we too hate counting and try and avoid it as much as possible. Other types of things we try and understand are when pollen is being released or how it moves through the air. I think that's most of that project. There's a lot of cool things about the intricacies of the remote sensing we are using, but if you want to know more about it, you'll have to come stop by my lab, say hi. We're up in Bradfield. Looking forward to talking with you all more. Thanks.
The question, well, I will make an observation. So I think what's exciting to see some of this wonderful work, urban plant science, right? And we've just hired a new urban plant ecologist, Karen Sexton, who will be joining us very shortly. And I can see already all sorts of threads that can connect him with you, and also Justine and so on. So we're sitting here and listening to these talks, right? Jean-Luc.
This is a question. Do you also have models with a bunch of parameters?
Sometimes it depends on the type of model. We try and do some model. So basically, a lot of this is trying to create predictions. And so we are often okay with throwing a lot of things and seeing what sticks. Generally, we do try and think about what is most biologically meaningful and take out the things that don't have much predictive value.
Okay, thanks.
Thanks, y'all.
This has been a production of Cornell University, on the web at cornell.edu.